as well as the aging study, more importantly, we did a major study in mild to moderate Alzheimer patients. This was done in the States, in the US, in Texas medical school. So this is done independently of myself as an academic. So it was I think what we're talking about it was around about 70 or 80, N equals 70 or 80 individuals. It was a sham double-blind sham controlled trial. So when I say sham, what the individual had was a helmet, basically, looked like the real one. But when the button was pressed, the light, they didn't receive any of the infrared light. The beauty of infrared light is invisible, so you can't see it.
Right.
So you can do a true sham controlled trial. And the beauty of the trial also, it was done at home, so it was you basically, it's not like you have to go into a hospital or take a drug. You basically can treat yourself at home, obviously with the help of a carer.
Right.
So that trial showed remarkable results, actually. I mean, it showed basically a range of memory performances, and the major one was the so-called mini mental score assessment, which is an assessment of between 0 and 30. Basically, most of us hopefully are in the high 30s, up that end of the scale. When you're in the mild to moderate Alzheimer's, you're down in the low 20s, high teens in terms of the number. And what we found with the trial was a remarkable nearly 5-unit improvement over 8 weeks with no side effects, which is unheard of, basically. As I said, this was sham controlled. In the sham treated group, there was, an insignificant change.
Hello, Dr. Chazot. You are a professor at Durham University, where you study rational drug therapies to combat major neurodegenerative diseases. So welcome to Modern Healthspan, and thank you so much for joining us today.
Hello. Yes.
So
thank you for the invitation.
You're very welcome. So Dr. Chazot, could you give us a little bit about your background? Because I believe you started in neurology, but what we want to talk about today in this interview is really about red light therapy. So how come you kind of transitioned or included red light therapy in your studies?
Sure. Yeah. So as you say, I'm a neuroscientist, a neuropharmacologist. So I've been interested in the brain for many, many, many years, and I actually am a pharmacologist of what I call the Jim Black approach: rational drug design, understanding your disease, and developing a drug that actually, rationally combats the problem, and then coming up with a drug that would do what you need to do in a particular problem. In his case, it was blood pressure. How do you combat blood pressure? He basically studied how our blood pressure is controlled. High heart rate was, underlies high blood pressure, and he developed the beta blockers, so-called beta blockers, to block that high blood rate. So it's rational drug design. So that I've used that all my career, and in this case, contrary to I guess blood pressure, the brain is much more complex. So you need actually to understand the brain, and it's to a certain extent understand what's going on in aging and what's going on in age-related brain diseases. And it's a complex mixture of problems and stresses that you need to combat. You need to combat all of them actually to actually make any difference really in developing a therapy for brain-related diseases. So that's the rationale, understanding the disease first, and that's what I spent maybe 20 years of my career is understanding the brain, what's the problem, and then thinking about, can we come up with different rational strategies to make a difference in the brain. So it's what I call the 3 N's. You need to basically give you neuroprotection, neuroplasticity, and neurorepair. So you need to basically be able to protect the brain, the neurons in the brain that undergo the brain transmission, the signals in the brain. You need to understand. You need to do something about the adaptability of the brain, the so-called plasticity of the brain. You need to make sure that the brain can adapt as you age and as you go into a brain disease. And ultimately, you need to maybe even repair the brain. Actually regenerate the brain, regenerate the nerve cells that have been lost. So you need the 3 N's to actually make a real difference in brain disease. So that's what I've been doing, and I've been using different strategies, both drug approaches, there's classic drug approaches, but more recently, well, I say more recently, in the last 20 years, I've been interested in actually using light therapy. And light is a very important process in terms of what its effects on biology, and it's the whole range of the light spectrum that is actually important. So in the visible end of the spectrum, you need to, it's very important in circadian rhythm, and every cell in your body undergoes circadian rhythm, and it's controlled during the day and night, and the biology is controlled during the day and night by the visible end of the spectrum. And then you move into the infrared end of the spectrum, which is actually invisible. That actually, it turns out, is incredibly useful as a potential therapeutic, and actually does a lot of good to the body as well. And then you've got the UV end of the spectrum, which you may think is it's a damaging end, but actually it is actually quite important. Also biologically has positive effects. You need a certain amount of UV to be able to generate certain vitamins, vitamin D again. So the light generally is an incredibly important process, and the idea of photobiomodulation, the use of light to biologically modulate the system, goes across the whole spectrum, as I said. But in terms of therapeutically, the infrared range is it appears to be pretty useful biologically, and that's what we've exploited for the last 20 years. Try to understand.
Right. So you're thinking about rational drug for well, I guess rational therapy design for the brain.
Yep.
So my understanding was that like Alzheimer's and Parkinson have different pathologies. How common are they? How much commonality is there that you can develop a single therapy, or are you targeting one particular disease?
No, in this case, actually, across all the brain diseases, there is quite a lot of commonality. There's general principles of stress, oxidative stress, inflammatory stress, and so-called ER stress, protein misfolding stress. There's different types of stress that affect cells, and including neurons, including the glial cells, which are important cells in the brain as well, which we shouldn't forget. And they affect, they are a part of all of the diseases. Parkinson's, Alzheimer's, motor neuron disease, all the major neurodegenerative diseases have a degree of oxidative stress, inflammatory stress, ER stress, and also in terms of common, and there are mechanisms that actually overlap as well between the diseases. For example, oxidative stress can lead to inflammatory stress, and inflammatory stress can, they jump around in terms of the stress of the cells, and how we cope with the cells depends actually. I mean, age is a big factor. As you get older, you can't cope with those levels of stresses as easily as you do when you are young. And each of us have our own version of these diseases as well, because we have our own ways of coping with stress built into the cells, built into the system, built into the brain. And so, yeah, there's a tipping point, basically. As you age, you tip in, you tip from the stress going on throughout your aging process. And then there's a tipping point depending on the individual's ability to cope into a disease. And that can go into Parkinson's, can go into Alzheimer's, can go into motor neuron disease. And like I said, that they have very common diseases. So it's actually quite easy to think of a therapeutic that actually could benefit across the board. Once you understand that you need a therapeutic that does the three ends, it does combat the complexity of the disease. You can't target brain disease with a single target, which has been the problem for many, many years, actually. In drug development, people have just focused on one thing, like, for example, in Alzheimer's, the amyloid story, they're just focused on that. That is not the whole story. In fact, it's quite a minor part of the story, actually, as it turns out. So, you need to understand that complexity requires a complex process. Unfortunately, something like infrared light of particular wavelengths appear to actually positively modulate those stresses. And it does pretty much all of the key stresses that you see in a brain disease, and also in aging as well, actually. So, yeah, that it's a complex disease and these complex responses, and we're just fortunate, yes, the powers that be has given us, a means of combating those complex diseases, which is all around us, basically.
Are you getting enough magnesium? Magnesium in the soil has been drastically reduced since the 1950s and is also removed by food processing, which explains why it's estimated that up to 75% of the population may be deficient. And if you've recently engaged in any sort of restricted diet for weight loss, there is a higher chance that you're not getting enough magnesium. Magnesium is a crucial mineral that's involved in over 600 biological reactions and offers amazing benefits for sleep, stress management, and hormone balance, all key factors for keeping your weight on healthy levels. Don't let magnesium deficiency derail your goals. Try Magnesium Breakthrough today and give your body the support it needs. Unlike other supplements, Magnesium Breakthrough by BiOptimizers contains all 7 forms of magnesium that your body can actually absorb, ensuring you get the full spectrum of benefits. For an exclusive offer, go to bioptimizers.com/modern and use the promo code MODERN10 during checkout to save 10%. And if you subscribe, not only will you get amazing discounts and free gifts, you will make sure your monthly supply is guaranteed. If we took Alzheimer's and so if you think about, because I believe that was the one that you studied first, like the specific pathology of Alzheimer's, and how does red light combat the different kind of elements of that?
Okay, so I mean, Alzheimer's disease is basically a process. I mean, actually, humans are the only species, maybe a couple of the primates on Earth, that actually develop Alzheimer's disease. It's unique to us, and that's an interesting.
Yeah, I was interested.
Mice, rats, rodents, other animals do not develop Alzheimer's. They develop their own version of dementia, memory problems, but they don't actually develop the disease. So it's sort of unique to humans, which is a really interesting concept. So what actually causes, that process to happen? Like I said, it's a combination of stresses. So again, it's an age-related process. I mean, you can develop Alzheimer's when you're young, but it's very rare. But generally, it's an age process. So there's, as I said, as you age, you lose the ability, or you reduce the ability to cope with the stresses, especially depending on your lifestyle, how you nutrition and exercise and all those sorts of things. If you don't look after yourself, again, you'd be sort of pushing your luck in terms of tipping into the disease. So Alzheimer's disease is a series of stresses. Oxidative stress, a combination of inflammatory stresses, does happen quite early on. So you're releasing inflammatory, pro-inflammatory proteins into the body that cause harm. Basically, it kills neurons, it kills glial cells, and there's a process called senescence. So senescence is what happens in your body generally, but if you're thinking about the brain, senescence is a process where your cells have to make decisions as you age. You can reach a decision where a cell can start to replicate too many times, and that can take you down a sort of a cancer route. Okay, or it can go down a senescence route where you actually stop dividing. So cells stop dividing, and they then turn into a cell that starts releasing these nasty inflammatory cytokines. And they do that at relatively low levels, but chronically, long term. So you're constantly releasing these cytokines into the system, and they start killing neurons. So you in life, you go down one or two pathways. Basically, you either over replicate, the cells divide too many times uncontrollably, can cause cancer, or you can go down this senescence route, which actually results in cells that ultimately, will kill neurons. You start to lose neurons, you start to lose connectivity, you start to lose ability to adapt, and things like that. So senescence, we think, is that sort of decision making, and maybe it's the tipping point to disease. That's still, not been proven, but my feeling is that this senescence process is your tipping point, whether you go down this process where you are starting to produce inflammatory markers, you're starting to stress cells, what we call oxidative stress. So your mitochondria, which are your batteries in your cells, are starting to not function as efficiently. So you're not producing sufficient energy to maintain a nerve cell's function. So it's a combination of those sort of things. And the third thing is actually what we have normally in our brain, and this happens actually when you go to sleep, is basically your brain deals with proteins within the cells that you don't want. These are unwanted proteins, and they get disposed of through what we call a lymphatic system. So at night, this happens. And again, if you don't get a good night's sleep, again, that's something to bear in mind. Again, everybody is different. That process, it isn't carried out efficiently. So your protein, these unwanted proteins stay in the cells, and they start to aggregate, and they start to then obviously cause the cell to be stressed and ultimately to kill the cell. So that I'm talking about these amyloid proteins in Alzheimer's, or the alpha-synuclein proteins in Parkinson's, or tau proteins, for example, in motor neuron disease. So these proteins are normally cleared from the body, but again, as you age and as your sleep quality deteriorates, that becomes a stunted process, and it takes you into a disease situation. And some cells are more sensitive than others. So, for example, in motor neuron disease, the motor neurons are more sensitive to these stresses, and that's why you lose motor neurons in motor neuron disease. Similarly, in Parkinson's, in some people, the dopamine neurons that are lost, those neurons are actually quite sensitive to stress. They don't have so much, what we call antioxidants in the cells, so they can't cope with stress, and they ultimately die. So you start to lose those neurons, and that results in you developing something like Parkinson's. And similarly with Alzheimer's, it's other types of cells that are important in memory processes. So again, it's the disease is a series of steps, or they're actually run in parallel. There's oxidative stress, inflammatory stress. As I said, this protein dealing with unwanted protein stress, so-called ER stress, is starts to deteriorate. And the beauty of the light therapy, the infrared light therapy that we developed the idea of using, and it's a specific wavelength. That's the first thing to bear in mind. Wavelength we use is around the 1070 nanometers, which actually 20 years ago was thought to do nothing biologically, and therefore we actually had to convince a company to make the LEDs to use it. And the reason we chose 1070 is because of that range of wavelengths. was that it penetrates the brain. The first thing is you need to understand when you're a drug developer or a therapeutic developer, your therapy has to reach the targets where the problem is in the brain. And the brain is quite obviously, it's a structure underneath a skull, and it's quite large, and you have to penetrate. And light, again, people think how can light possibly penetrate tissue, but actually the infrared ranges can penetrate quite significantly, and how the wavelength we've adopted and used and studied for many years is the most penetrating. It turns out to be the most penetrating wavelength in the infrared range, and that's a good thing. So obviously that suggests that it will get to your target where you want to treat a disease. And the other important thing of this wavelength is the peak wavelength for transmission through water. And water makes up 70% of your brain. So once it's in, it actually travels further into the brain because of that fact. And that's the reason we chose that wavelength initially. But as it turns out, okay, it gets to target, but also it does all the things we wanted a therapy to do in the brain. It does pretty much everything we've just described for the disease, like Alzheimer's. It's an antioxidant, anti-inflammatory. It actually targets this system that clears unwanted proteins. So one of its targets is a channel that controls that process in the lymphatic system. So that's one of the advantages of this wavelength is actually it targets that as well as the other major target, which is the mitochondria. So what it does, it boosts mitochondrial function, which is well known for many of the wavelengths of infrared light. It boosts ATP production. It boosts a very important thing. It actually increases a little bit of nitric oxide, and this is a gas. You might think it's a bit strange, a gas, nitric oxide, but actually nitric oxide is an incredibly important molecule in your brain and in your body generally. It actually controls whether your blood vessels dilate or not. So what it does, it relaxes a blood vessel and increases your blood flow in the body, and that's what our light, the light therapy does. The infrared light therapy, many of the wavelengths do this. Actually increases a bit of nitric oxide, so locally your blood supply is much stronger, so you get more blood flow, more oxygen to the brain, which is very important in aging. It's very important in brain disease, because again in aging, your blood supply starts to wane a bit, so you don't get so much oxygen into your brain, which is not a good thing, because the brain neurons need oxygen. In fact, they use about 35–40% of the total oxygen in your body, because they are very what we call excitable cells. They need to function and be excitable, and you need energy to do that. So ATP and energy production is incredibly important. And fortunately, like the infrared light therapy does give you that extra blood supply. It actually increases oxygenated hemoglobin, which is the very important carrier of oxygen to your brain. And again, 1070 seems to be the optimum wavelength for doing that. It gives you a longer extent of oxygenated blood to your brain, which is all good stuff all good news for aging. It's all good news for age-related diseases where it starts to deteriorate. So yes, so as well as that, one interesting thing also is it also in the mitochondria, what it does, it produces a little bit of what we call reactive oxygen species, which most people will say, oh, that's not a good thing. This is an oxidative stress molecule. But actually a little bit of oxidative stress is actually a good thing. What it does, it triggers your body's system to protect your body. So it's what we call preconditioning. And the light appears to do this. I call it sort of photo preconditioning. So it does produce a little bit of reactive oxygen species, which then causes transcription of key protective proteins, and these are called chaperone proteins. So we've shown with our wavelength, again, in an Alzheimer mouse model that you can actually produce, you increase these so-called chaperone proteins, and these chaperone proteins are really important for protecting your cells from this stress that I've talked about all the way through, all these stressors. So it protects against oxidative stress. It helps with, again, with the protein stress, the misfolded protein stress, and also, yeah, it processes those misfolded proteins. And what, you know, but the biggest result we found a number of years ago, what I call a fall off the seat moment, was actually treating the cells of our light, increased the chaperone proteins, but at the same time, what it did, it reduced amyloid, one of the key proteins in Alzheimer's disease, down by 80-odd%. It was a massive reduction in amyloid. And it wasn't just the amyloid that you find on the outside of a cell. It's actually in the key amyloid you need to target inside a cell that's killing the cell ultimately, was reduced by this therapy. And also we found the other key pathological protein, phospho-tau, was again reduced by this treatment. And we think it's because of this preconditioning process that the chaperone proteins are reducing these unwanted proteins that are in the cell. And that was obviously a really exciting observation in our preclinical studies, that, you know, that you could have a treatment that would actually reduce the key pathological proteins within Alzheimer's. And that led to the first trial, you know, actually in humans, you know, the idea that we could actually potentially use it in humans for treating Alzheimer's disease. Okay, so it's a combination of things. It's the oxidant, you know, all the stresses we talked about, it combats, and it also importantly reduces some of the pathological proteins as well, and no side effects. That's, again, I have to emphasize.
Yes. So once the amyloid has formed into a plaque, which it does on the outside, can that be got rid of, or does that remain? You're just stopping any more being formed?
To be honest, that's too late in the process.
Right.
Yeah, the plaques are the last process, part of the process. What we need to do, again, to combat any of these neurological diseases where misfolded proteins are important, is that you need to start treating earlier, as early as possible, actually where at the point where the proteins are starting to produce what we call fibrils, oligomers, fibrils. That first of all, they're just a single protein, then they start to form oligomers, strings, then fibrils, and then ultimately they're kicked out of the cell and they produce plaques. So that's the last process. So these treatments that are out there, unfortunately, they're treating the disease too late. You know, but if you start put, you know, the proteins on the outside of the cells, particularly in Alzheimer's, the amyloid plaques, you know, it's too late in the process. The cell's already died. That's produced, you know, those plaques. So mopping up, you know, the external amyloid is, yes, it will have some effect. It will probably, you know, help a little bit down the line, but ultimately it won't do much to the disease progress. You know, it won't do very little. So that, unfortunately, this, you know, the current therapies that are in the press and in the media a lot, and, you know, caused the FDA panel to resign in the States because it was passed through, and NICE have approved it in the UK. Those processes, to most of us, you know, are really red herrings. You know, they're not going to cause, they're not going to, you know, make any real major clinical difference. Whereas treating our individuals with the light therapy as early as possible will, you know, stop that, you know, reaching that point, if you like. Will actually protect the cells before, you know, any problems later. So yes, what we showed preclinically was that it reduced the amyloid inside the cell, you know, the actual amyloid that's actually killing the cell in the beginning of the process, as well as the phospho-tau. So that was really important. I mean, we also, we have shown in the past also that it improves memory performance and in premature aging and things like that. That was our first study. We showed that process going on. So, in other words, the therapy is relevant to aging. In an healthy aging individual, it will potentially help. Help in terms of the tipping point, if you like, pushing the tipping point back time. That would be, the thinking. And we have done a small study in healthy aging individuals and shown improvements in memory performance, even in healthy aged individuals that didn't have, official dementia. They improved their sleep. Actually, we didn't publish that, but anecdotally sleep was improved, which goes along with the idea that sleep is really important in aging, the aging process, for that clearance of those proteins, for laying down memories and for actually dealing with your mental health. Sleep is incredibly important. And yeah, that in terms of removing unwanted proteins through the lymphatic system, that's really, again, really important part of the whole mechanisms underlying light therapy, to be honest.
Did you look at executive function as well? I mean, you mentioned memory, but did you look at executive function?
A range of memory tasks were done in the, so we did as well as the aging study, more importantly, we did a major study in mild to moderate Alzheimer patients. This was done in the States, in the US, in Texas, medical school. So this is done independently of myself as an academic. So it was I think what are we talking about? It was around about 70 or 80, N equals 70 or 80 individuals. It was a double-blind sham controlled trial. So when I say sham, what the individual had was a helmet, basically, looked like the real one, but when the button was pressed, they didn't receive any of the infrared light. The beauty of infrared light is invisible, so you can't see it.
Right.
So you can do a true sham controlled trial. And the beauty of the trial also, it was done at home, so it was you basically, it's not like you have to go into a hospital or take a drug. You basically can treat yourself at home, obviously with the help of a carer.
Right.
So that trial showed remarkable results, actually. I mean, it showed basically a range of memory performances, and the major one was the so-called mini mental score assessment, which is an assessment of between 0 and 30. Basically, most of us hopefully are in the high 30s, at that end of the scale. When you're in the mild to moderate Alzheimer's, you're down in the low 20s, high teens in terms of the number. And what we found with the trial was a remarkable nearly 5-unit improvement over 8 weeks with no side effects, which is unheard of, basically. As I said, this was sham controlled. In the sham treated group, there was, an insignificant change, basically, in memory performance. So that was a remarkable, first start. I mean, it's a small trial, relatively small trial, but it was a remarkable start. And since then, this was done 3-4 years ago, it was published. A number of other trials have used our wavelength and other wavelengths of infrared light to show, again, improvement in memory scores, executive function based memory. Different types of memory assessments the evidence is growing significantly across the board, and different types of memory as well has been looked at. And so that's an ongoing build up of evidence, if you like, to back up what we're talking about. I'm currently putting ethics together to run a similar, basically the same trial in the UK. Fortunately, I was able to get some funding to run a trial in the UK, and that will be the first trial to be run in the UK. Actually, we are a bit behind, more than a bit behind the rest of the world, actually. So there's many trials in, say, Australia and the US going on with a range of wavelengths, including our own, showing, again, the evidence base. I think I did a quick literature search recently, and I found about 25 papers with our wavelength range being used and showing positive effects. Both, many of them were clinical, many, and also preclinical studies. So the evidence is building, and I need to emphasize also, during the first lockdown of COVID, I suggested that this approach would be useful for long COVID, particularly the brain effects of long COVID, which are brain fog, memory problems, etc. And I again, it was a rational thinking. All the effects we talked about were relevant to the disease, and in fact, this actual wavelength that we're talking about was first used 20 years ago to treat cold sores. So herpes simplex, so it's an antiviral approach, and in fact, it's an NHS approved treatment for cold sores, even now still. So the initial use of this particular wavelength and this particular treatment was actually as a viral treatment. So as it turns out, yeah, a clinical trial was done recently in long COVID to show, again, positive effects of this wavelength for the various reasons we talked about. So it's also an antiviral approach, and again, some of the mechanisms we talked about, some of the chaperone proteins, HSP70 is one of the chaperone proteins, is an antiviral mechanism. Nitric oxide is actually an antiviral, also does modify viral replication. So there's various reasons why the light therapy would be useful for long COVID brain fog approaches. So yeah, so that gives you a bit of a flavor of some of the things that are going on and why, I guess.
Right. So kind of looking at the light in a bit more detail. So we've talked about the wavelength, although I would like to kind of think about that a bit more, because I did actually try to find any devices in the 1070 range. And I think I found like one company, but the vast majority of devices that are available commercially are like 810, 850.
Yep.
What difference do you think? Can you comment on whether there would be any what differences you would expect?
Yeah. So just the first point. Yeah. So I mean, there are two companies basically that you can get the wavelength, including my original clinical colleague that knocked on my door 20 years ago. That was used for the trials I've been talking about, actually. Anyway, but actually some companies are starting to look at 1070. But going back to your original question, the wavelength, the two main wavelengths that I think are most useful are ours, obviously, but also around the 810 nanometers. And the main one of the reasons is, first of all, it penetrates, similar to the 1070 into the brain, so it gets to target pretty much equally to the 1070 wavelength. So that's the first point. And also what it does, that particular wavelength, yes, I would say is potentially very well, it is very useful. There is clinical evidence for its use, and it does similar things to our wavelength. So around about the 810, I would say is a very useful wavelength. In fact, we're very keen to compare it. We're currently working on comparing the two wavelengths, and we are working with a company that has worked with the wavelength a lot. And in fact, that company also is looking at exploring combining 1070 with 810 in a dual system, and it's doing it actually quite cleverly, because it may be that you need to target one of the wavelengths to a certain part of the brain, whereas the other one potentially is more useful subcortically versus cortically. So that's been looked at, and what's this space? Basically that will be available, and there's some papers coming out on that idea of doing a combination of the two wavelengths in two different parts of the brain. Actually, the other thing I haven't mentioned actually is the physiological effects of this treatment. A lot of EEG has been done in the brains of people that have been treated. Again, you see modifications and plasticity of the brain. The physiology of the brain changes positively with the treatments, and both 810 and 1070 do again do similar things. So I would say 810, all in all, from the evidence, is as useful as ours. The only thing is what we don't know, because the research hasn't been done, is whether the 810 also targets this lymphatic target I mentioned. So it certainly modifies the mitochondria, boosts ATP, oxygenated blood, all the things we've been talking about. Nobody studied the chaperone effects, which we have observed. That's another thing we don't know that the 810 does. My guess is it will do similar things. But also, as I said, we don't know if the 810 actually modifies this lymphatic effect, which I think is incredibly important. And it's a sort of benefit of the wavelength we study. It targets the mitochondria, and it does this lymphatic process. And you need both really to give you maximum benefit in a disease like Alzheimer's. So yes, in answer to your question, 810 is a good penetrating wavelength. It does many of the things that the 1070 does. There's still some research that needs to be done that it does the full complement, if you like, that we've seen with our wavelength. So I mean, there are some successful clinical trials. There's small clinical trials I've seen with 810. And mostly, a lot of them are case studies, single studies rather than full studies. Like we've done a couple of full studies. So yes, 810 is fine. I mean, there are other wavelengths, and there are other companies that are selling devices that give you other wavelengths, shorter wavelengths. Again, those wavelengths have some logic, in that they again will do a lot of what we talked about in terms of the mechanisms, positive mechanisms, particularly on the mitochondria effect. The only problem for me is penetration. The shorter wavelengths really don't penetrate anywhere near into the brain. So they're useful for surface effects, maybe effects on the blood supply locally, and there is again, your viewers may not be aware, but actually it's recently found that you can find mitochondria, those batteries I talked about in cells, you can find them in the blood supply. So there's mobile mitochondria that basically travel around your body. So there is a possibility you can treat with these shorter wavelengths that can modify the activity of these mitochondria that then can get to places in the brain. That's a possibility. As I said, I'm confident with the 810 region. Again, there's a lot of they use 850. Again, don't get hung up on wavelengths. This when people say 810 or 808 or 820, and we talk about 1070, 1072, 1064, they're all the same basically. The thing is, people have just to give you some insight, there's an issue about patenting these devices and wavelengths. There's a lot of, they manipulate the number. People call it 808, 810 to avoid being issues of patent, issues of IP and things like that. So we've had to do similar things. Our original wavelength was 1072, and then we shifted it to 1070 and then 1068 just to avoid issues problems. I mean, it's very strange, and people,
Yeah.
the general public, don't realize it's basically the same wavelength. Don't get hung up on, oh, 808 is different to 810. It's a number. It's plus and minus 10 nanometers, 10, 15 nanometers is basically the same wavelength doing the same thing mechanistically. And there's a common misconception. I get asked a lot about that. And also there's things about, again, some devices give you a constant light, whereas we think pulsing, it might be a question you might be asking me next, pulsing is important. So pulsed light and also the power of the light for penetration is important. So again, you get variability within companies and devices in terms of that as well. And again, using LED or lasers, again, we favor LEDs more on a safety side of things. But saying that, people had sell devices with lasers, and they have published reasonable evidence for use. So there's things like that. So pulsing is important, and the power. So you need a certain degree of power as well as for penetration. And if you haven't got enough power, really, again, your penetration is limited. So and pulsing we think is important and not repetitive. Right from the beginning, when we did our preclinical studies, repetitive treatments is important, not just a single treatment. All our preclinical was a series of short but repetitive treatments seem to be beneficial to the process, to give you sort of the best results, if you like. And we do similar things, obviously when we're treating humans, it's twice a day, 6 minutes, short exposure, twice a day, 6 to 10 minutes, every day. Again, it's repetitive treatments seems to give, well, it gave us the results we got. I wouldn't say, I can't claim that that's optimal by any means. We haven't had the finances to basically to do all the experiments, basically to demonstrate that we have reached optimal levels of treatment. But as I said, we've got a protocol that works, appears to work, and we've stuck with it up to now. But there are ways you can improve it, and that's the next level, if you like, of this process is to tie it in with what we call biofeedback processes. So you can treat people with the lights, but you can also do some biofeedback, which involves again playing around with the physiology of your brain and targeting the treatments as well, thinking about where you need to target. So everybody's different, as I mentioned right at the beginning. Personalize your treatment, find out actually where the problem is in an individual brain. You can do what we call an EEG, so it can map your brain, and then also design a device where you can selectively maybe target that part of the brain more than other parts of the brain which are working normally. So there's aspects like that can optimize the process. So the light therapy can basically give you a certain status, if you like. There's means, because of the personal nature of the diseases, there are ways that we potentially can improve and optimize the process to really make sure that you are slowing down the disease, or even getting to a point to stop the progression. So that's the ultimate, obviously, holy grail, and that's where we are at the moment. As I said, we're quite far off being optimal, but it's at a point that we seem to get some very strong positive effects.
Can we just go back to power? So you said you need power. You need a reason. How do you quantify that? And what kind of power are you looking at? I mean, I guess it's in terms of milliwatts per centimeter squared or something like that.
Yeah, sorry. Yeah, you've got me on that one, actually. I couldn't give you the number specifically that we use, but yeah, you need a certain quite high degree of power. I mean, actually, that generates heat, and that's actually
Mm.
part of the helmet design, that we have a cooling system.
Yeah.
But yeah, so I mean, in terms of the physics and the specifics, what we can do is, if your listeners are interested, I'm very happy to give you some specifics after this
Okay.
presentation. Yeah, I mean, I don't want to give you a false number,
Right.
as I said.
Yeah.
Yeah, I mean, again, my colleague who's actually built the device has been tweaking it recently, so I can give you the up-to-date sort of idea. But the main thing is that you need quite a significant amount of power that actually to get the penetration.
Yes. Yeah, because there are hats you can get, but I think they're mostly for hair, and they just don't have the power. They're not
Yeah,
that.
absolutely. Yeah, there are some devices out there that really, they're very cheap,
Yeah.
but they're just not they're not giving you sufficient power to get penetration.
Dr. Chazot provided a few details for the light used in the studies that he confirmed after the call. In the interview, he mentioned a wavelength of 1070 nanometers. To confirm, the wavelengths used were from 1060 to 1080, and as he refers to in the interview, the exact wavelength may not be that important and is mostly driven by IP issues. The overall power of the device is 15,000 milliwatts, which is 15 watts. This gives a power density on the skin of 23.1 milliwatts per centimeter squared, across an area of 650 centimeters squared. The device does sit on top of the head, so is very close to the surface. So we did talk a little bit about what the next steps are. So you are doing some human trials. Do you have any idea when this would be in the clinic? I mean, how close are we to having it available?
That's a good question. I mean, again, I think it depends on the country. At the moment, it's every country is different.
Mm-hmm.
In terms of their acceptance of the approach. Like I said, there's enlightened countries like Australia and the US that are prepared to trial it and work it through. Unfortunately, the UK is, like I said, well behind in the process. So, in terms of the what we're ultimately aiming for, it's certainly in the UK, is to have an NHS approved device that can be used routinely. We're a little way from that, I would say, because obviously we've got to do the trials, and also we've got to build up the confidence of NICE for this process. Whereas in other countries, like particularly in Australia, for example, that they are definitely, they could be seriously using this in the next year or two. We're talking very short term. And America and the US again, they will be looking at this before we will in the UK. I mean, the thing is, the devices are available. So actually, for people with who want to look at it in terms of their normal aging, or health, or if they have a disease, that they can purchase the device, or the devices now. So in terms of for general use across the public, yeah, I think certainly in the UK we're looking at least 4 or 5 years, to be at that acceptance point. I think what will happen, once we run this first trial, things will go like that in terms of the UK. If we can basically get the ethics and we can run the trial, and the assumption is we get the same results we got in the US, people will start to wake up and actually see, and things will move a lot quicker. And yeah, because it's sort of a no-brainer, because they will realize, the current therapies that are coming through, there are one or two useful things other than the amyloid drugs, which will die a death very soon. There are one or two other useful things coming through, including, like I said, I've talked to you before this session. Now we have a drug regime that is looking very promising. So things will start to move generally in treatment of Alzheimer's and neurodegenerative diseases relatively quickly in the next 2 or 3 years, and light therapy hopefully will be part of that. Again, it's just to get over that initial, I don't know what it is, risk averse attitude to light therapy, skepticism. It has a long history. It has a checkered history over the years. But, at the end of the day, when they start to be evidence is building over the next year or two, they will have to take note at the end of the day, because, to have a treatment that gives you any sort of clinical benefit with no side effects, relatively quick effects, easy to use at home, then it's sort of, it should come. I'm hopeful before I retire that this would be taken seriously, certainly in the UK, because
that certainly is very optimistic, given how little progress has been made in the last 25 years or more.
Absolutely, yeah. No, we're in actually generally, not just with the light therapy, we're in a quite a good place. There are a number of, again, rational things coming through. Some of them are actually what we call reuse, i.e. drugs that have been used for other things, particularly say in Parkinson's. There's a couple of drugs that one was a diabetic drug, and there's a couple of things that are coming through that look really promising and gone into phase 3, and they make a lot of sense to me as a rational pharmacologist. So I think I am very optimistic, actually, that in one way or another, the light therapy or together with rational drug therapies will be coming through. But we have to get away from this unfortunate, the first drug that really people have started thinking that we can solve Alzheimer's. They will be disappointed, unfortunately, and a lot of money has been spent on it. I shouldn't say that, obviously. The companies won't be too happy with what I say, but I'm not the only one.
Right.
Unfortunately, people have died on those trials, which is not good.
No. We
need to rethink, and just like I said, we need something that would cover the whole gamut of mechanisms that underlie these complex diseases, and light therapy is one of those. And there are one or two drugs coming through that seem to be targeting the right in the right way.
Right. So we are a little bit over, but so one.
Sorry.
Yeah. No. One question. So you talked about being able to regenerate neurons. But I mean, and one of the problems one of the outcomes of Alzheimer's is the neurons are dying, right? So if you catch it late, there's like no hope, because if the neurons are already dead, then so is there any hope that we could be able to regenerate some of the neurons that have died?
Okay. Yeah. If we're just focusing on the light therapy, at the moment, I have to be honest, we don't have that evidence really, but for that a third of the 3 ends, if you like, the repair. There is scope, and there's potential, and there's what we do know is that it does stimulate production of what we call neurotrophins, particularly BDNF, brain-derived neurotrophic factor, and one of that's a really important neurotrophic factor, which actually is involved in plasticity in the way the brain adapts to stresses, but it also is important in regenerating neurons, making neurons actually produce processes, and actually expand and make new connections. So light therapy does increase the production of that neurotrophic factor. So that gives us some hope that potentially it could be useful for neuro repair. But I have to be honest, we haven't done a lot of work in that part of the mechanisms, in terms of demonstrating that it will give you regenerative process. Other people have started to look at it, and I've started to produce some evidence, but I would say that's the weakest evidence base at the moment for that part of the process. I mean, the major thing to bear in mind for this process, you need to use it as early as possible. Then you don't have to worry about repair. At the end of the day, what it does do, and it does it early, can do it early, is to slow things down, keep neurons happier, make new connections. We know all that. So at the end of the day, that's really important part of the process. If we can, that's another thing my lab works on is early biomarkers, and actually working out that somebody is going to be going down that path as early as possible. Then obviously we can intervene either with a light therapy or with a drug as early as possible. And if you can do that, you've got a much better chance of putting off the inevitable, if you like. So in answer to your question, the light therapy, I think, is most useful to be used as early as possible. Then you don't, like I said, you don't have to necessarily worry about the repair, because you're actually protecting the neurons in the first place, and you keep maintaining the connections in the first place. So down the line, you've got less chance of it being needed.
Yes.
Yeah. But there's more research needs to be done, certainly with light therapy and neuro repair, to demonstrate it will be beneficial for that part of the process. But as I said, if we start treating people earlier, the better.
I mean, it kind of since there are no side effects, and it helps with the brain, with the normal cognition. For an aging brain, it seems to make sense. You just do it anyway. You get to like 50 and you start wearing a red helmet.
Potentially, that's right. Yeah, I mean, particularly in individuals that have a familial history of brain disease. So if you have a genetic disposition potentially to have problems in later life, certainly those individuals would benefit from an early intervention. So just build it into your normal sort of exercise routine, regime. 6 minutes twice a day, plus your exercise. You need to do the other things as well.
Yes.
It's not like the complete magic bullet. I think you build it into a sort of a normal, sort of healthy regime. So exercise, sleep, good sleep quality, plus a bit of light therapy to help you along. I think it's the combination of things. Good nutrition again as well. Obviously diabetes is a major issue. Overeating the wrong things. So I think it's part of a healthy lifestyle regime when you reach a certain age would be sensible. But as I said, particularly in individuals that have a family history, again, of disease, and there are people, there's about 5% to 10% of the population that have a genetic, the chances of developing any major neurodegenerative disease is much higher. Those individuals would obviously potentially benefit from this therapy.
Yes,
that's what I'm saying.
Absolutely. And lifestyle always comes first. We definitely
absolutely.
talk about
Yeah.
that as the
Because you need
main...
also, you need to, thinking about the visible end of the spectrum as well. You need to, circadian rhythm needs to be good. You need to get out fresh air and light, as Florence Nightingale said over 100 years ago, is the two main things really for health and well-being. That's how she dealt with the Spanish flu. Basically, she wheeled the patients out into the sunlight and the fresh air, and many of them recovered, basically.
Right. Excellent. Dr. Chazot, thank you so much for joining us today.
No
It's
worries.
been great talking to you.
Thank you very much.